So, well, good morning, everyone. I was chatting with some of the students earlier, and hope you guys are all doing well. And students look like they're hanging in there with all this virtual format. And I heard at least a couple of students saying they are learning new things, so that's good. And so today what I'm going to do is, so my name is Ram Dixit. I think I've interacted with all of you guys several times now through orientation, etc. So I'm not going to say more than that, other than maybe if it's been a while. My lab is a cell biology lab. I am interested in the cytoskeleton, which is the topic for the lecture this morning. And my lab specifically focuses on the role of the cytoskeleton in cell morphogenesis. So we do a lot of biochemistry and cell biology combined with some genetics because Arabidopsis is a really powerful genetic system. So that's really helpful and for sort of gene discovery and functional analysis. So we study aspects of microtubule cytoskeleton, how the dynamics are regulated, how microtubules become organized into specific structures, and what their purpose is in cells. So today's lecture, I'm going to start sharing my screen now, and I think is the recording resumed? You can't see. Oh, good. Yeah, it has. All right, so hopefully you guys can see my slide there, the first slide. Okay. So as I said, we'll be talking about the cytoskeleton. And the cytoskeleton is a pretty conserved system in both plants and animals. And in fact, for a long time, people thought the cytoskeleton was a unique feature or a distinctive feature of eukaryotes. And so in the older textbooks, you'll see eukaryotes and prokaryotes being distinguished by the presence of organelles in the eukaryotes and also the cytoskeleton. That was sort of one of the features that people listed, and that is certainly not true. It's been now known and appreciated for a long time now, close to 20 years or so probably, that prokaryote, well, at least 10, 15 years, prokaryotes have a cytoskeletal systems. There are analogs of actin and microtubules and intermediate filaments in various prokaryotes, and they perform roughly the same kinds of functions, in terms of conferring cell shape, facilitating plasmid segregation, things like that. But today we're going to focus on eukaryotes and specifically sort of a little bit of comparing and contrasting between plant and animal systems. And so I will say a lot of the images that I will show, unless you see some other credit given, are from the Tom Pollard's really excellent cell biology textbook. That's the one I use for my upper division undergraduate cell biology course here at WashU. And so a lot of these images are from that textbook. So in outline for today's talk, really, I'll just very briefly introduce. So this is an introductory talk, so we won't have time to really drill deep. And as for the other lectures, you guys have any questions, please just chime in. Don't bother with the raise hand or anything. Just unmute and say something, and I will pause at the next opportune moment to address your question or comment. So very quickly, I will introduce the major the components of the cytoskeleton, the actin microfilaments, or F-actin, microtubules, and intermediate filaments. We'll see a couple of brief examples of how their arrangement is similar or different between plant and animal cells, and then touch on functions of the cytoskeleton. And really here, we won't have time to go into all of the functions or even some of the major functions. So we will focus on cell morphogenesis and animal cell migration as examples. And you've already seen some of this in Amit's talk yesterday. He talked about focal adhesion sites and stress fibers, which are composed of actin filaments, and might have seen something. Probably you haven't seen anything much about microtubules so far. And Anders probably talked about actin also in his talk on forces in cells. So what's the cytoskeleton? It is a network of protein polymers. So these are polymers composed of protein subunits, which form filamentous structures. And the important thing that I want to emphasize here is that the cytoskeleton is a very dynamic structure. Okay, the discovery of the cytoskeleton was based on electron microscopy. And so, the name belies that, the fact that these structures looked like struts and beams inside cells, and so they look like a skeleton of cells, and so hence the name cytoskeleton, the skeleton of cells. But unlike our cytoskeleton, which is a relatively static structure, the cytoskeleton, the cellular cytoskeleton, is extremely dynamic, and the dynamics of these polymers are in fact very important for their organization and function and the ability of cells to adapt to changing conditions, right? And so the three components of the cytoskeleton are actin filaments, also called microfilaments, or F-actin for filamentous actin. So you might see F-actin on some of the subsequent slides, and that refers to the polymer form of actin or actin filaments, microtubules, and intermediate filaments. The cytoskeleton is really important for a variety of essential functions in cells, in both plants and animals. The cytoskeleton sort of provides internal structure to cells and is important for a shape determination of both animal and plant cells, but I will emphasize later in today's lecture how the cytoskeleton achieves its this task is very different between animal and plant cells. It organizes the cytoplasmic contents, right? So life is essentially fundamentally out of equilibrium condition, and so organelles and other cellular components are not just all uniformly or homogeneously mixed in the cytoplasm. It isn't just a bag of enzymes, as some of the older biochemistry textbooks used to refer to the protoplast. But there's a lot of structure inside the cell, and organelles are positioned and are repositioned as required. And a lot of that depends on the cytoskeleton. Okay? And a lot of it, not all of it, is dependent also on motor proteins that I will introduce toward the end of today's lecture. That move organelles and other cellular material along cytoskeletal tracks. The cytoskeleton, from a mechanobiology standpoint, is really critical for force generation in cells, also involved in responses of cells to forces. So you heard some about that yesterday when Amit was talking about maturation of focal adhesion complexes and in response to external force and so forth. So these polymers, as they grow and shrink, as they add on new subunits and hence grow in length or lose subunits and therefore shrink in length. And there are other ways of modifying these cytoskeletal filaments as well, like cutting them and so forth. Through these activities of growth and shrinkage and other dynamics, these polymers can exert pushing or pulling forces on organelles, for example. So can anyone just briefly think about or describe how a cytoskeletal filament might exert a pushing force? What is it about the filament that might produce or generate a pushing force? How could that be achieved? Anyone, just chime in quickly so we can sort of move along.
Extension of like filopodia into ECM.
Right. So it's the polymerization. As the polymer grows, there's it's going to exert a force on the plasma membrane, for example, from the context of filopodia or lamellipodia, and we'll see the example of dendritic actin-mediated sort of lamellipodium formation from the context of animal cell migration a little bit later in today's lecture. What's an example of a pulling force? How would that work? Could you exert a pulling force during polymerization phase as the polymer is growing? Which phase do you think is more conducive to it exerting or generating a pulling force on some other structure? Growth phase or shortening phase?
Shortening.
The shortening phase, right? So if you were to couple the shortening of a filament to some other structure, then now you can exert a pulling force, right? And an example of this, not quite a pulling force per se, but I mean, it is in a sense, is a famous example is, during anaphase as the sister chromatids are being separated, right? So that's where the sister chromatids, different organisms have different mechanisms, different between mammals versus yeast. but there are mechanisms to connect or couple the sister chromatids at the kinetochores to the plus ends of the kinetochore microtubules in a spindle apparatus. So as that microtubule starts to depolymerize from the plus end, those sister chromatids are sort of pulled towards the poles of the spindle, and so they separated, right? So cells harness the polymerization and depolymerization phases of the cytoskeletal filaments to do work, right? To exert force in the form of pushing or pulling forces. Now, a lot of other mechanobiology that is cytoskeleton dependent is driven by motor proteins. So these are proteins that actually walk along cytoskeletal filaments, particularly in specific actin filaments and microtubule tracts. There are no known motor proteins that move on intermediate filaments. And so motor proteins that are bound to the cytoskeleton can exert force on the filament itself or facilitate the transport of vesicles. And we'll touch on this also toward the end of today's lecture. And frequently, these are not mutually exclusive. As I point out in the last point there, you can have both polymer-driven forces as well as motor-driven forces working together. And really, a great example of that is mitosis. The spindle apparatus is really all just about force generation and force balances, where both polymer-driven forces and motor-driven forces are really important for the assembly and function of the spindle. So here are the components of the cytoskeleton. So we have actin filaments. These are the ones first discovered. They are about 6 nanometers in diameter. And on the top here, it's a diagrammatic version of these. And then on the bottom are some immunostaining or immunofluorescence examples of animal cells. So you can, you've seen, I'm sure, examples of these kinds of pictures in the past. So here we have actin filaments in the form of stress fibers. And then we have microtubules, which are tube-like cytoskeletal structures. So they're cylindrical structures. And famous one is the radial microtubule array during interphase, or the spindle apparatus during mitosis. Microtubules are 25 nanometers in diameter. And then finally, we have intermediate filaments that are about 10 nanometers in diameter. And here we're looking at keratin filaments extending from desmosomes in epithelial cells. So this is very important for skin and other kinds of epithelia, for example. So if we were to think about the mechanical, just some basic mechanical properties of these 3 cytoskeletal filaments here, actin filaments, intermediate filaments, and microtubules. Of these 3, which one would you expect to be the stiffest?
Microtubules.
Okay, why? What's the reason for that?
Because it needs to be the longest.
Okay, intermediate filaments can be pretty long. Some of these keratin filaments actually can extend from one desmosome to the other desmosome on the other end of the cell.
Looks at bending stiffly, so the microtubules is larger than others.
Correct. And why is that? It's not. Its bending stiffness is high. Why is that? Or flexural rigidity is high. Why is that?
The structure, the special structure, the tubule or the pipe.
Correct. Yes. So it's the second moment, the second moment of area, right? So this is like a pipe, very stiff, difficult to bend, and it has to do with the second moment of area. So which is essentially a way to describe the distribution of mass in a transverse section. Here, the mass is at the periphery, and that's what makes this a very strong structure, right? So if you take a certain amount of metal and you mold it into a rod, or you take the same amount of metal and mold it into a pipe, the pipe is a much stiffer structure, right? Much harder to bend. And that's the reason why microtubules are very stiff. And then in contrast, then actin filaments are like thin rods. And so they are much, much less rigid. Flexural rigidity of microtubules is sort of in the millimeter range. Actin filaments is micrometer range, much less. And so frequently in cells, you'll see actin filaments bundled or kept very short in order for them to play a mechanical role more effectively. Okay, so it's the idea of you take floppy structures, but if you bundle them and cross-link them, then you have the resulting higher order structure is more rigid. The intermediate filaments, as I will show you briefly later, they have a braided rope-like structure. So think, those steel cables that hold up suspension bridges. You have thin filaments, woven into sort of intermediate filaments, and those are woven into, eventually the full cable. And that sort of hierarchical assembly process that resembles sort of a braided rope makes these polymers very flexible, but extremely strong and really resistant to tensile forces. And so in skin cells, for example, if you have mutations affecting either the desmosomes or keratin, then you have the skin is very fragile, can't resist even relatively gentle forces like, if you scratch it or pull on your skin, there will be rupture. Okay. And so the intermediate filaments really are important for resisting those forces in these kinds of cells. While the actin filaments and microtubules are present in both plants and animals, intermediate filaments, at least the sort of classical intermediate filaments, are not present in plant cells. There are some proteins that sort of vaguely resemble intermediate filaments, but there's no evidence to date of anything using electron microscopy or other techniques to show that there are sort of bona fide intermediate filament-like polymers present in plant cells. Okay. So you can, for our purposes, intermediate filaments are sort of an animal cell characteristic. Okay, so now what I'm going to do is just very briefly introduce you guys to some of the basics of the actin cytoskeleton, microtubules, and intermediate filaments. Just tell you what the building blocks are and some of the key aspects of polymer assembly and dynamics. We won't have, I mean, this just actin cytoskeleton alone in my undergraduate classes is a 3-hour section, and that itself is by no means complete, right? So here I'm going to do this essentially 5 minutes each. So actin cytoskeleton, the building block or the subunit is a G-actin, which stands for globular actin. So all of these cytoskeletal structures, by the way, are great examples of macromolecular assembly. This is a concept that Paul Janmay probably introduced, which is, you take what cells do is they build large structures, not as a whole, as a single unit, but rather it's like a Lego idea. Okay, you have lots of building blocks and you can assemble those building blocks in lots of different configurations to make different types of structures, right? So cells recycle these building blocks, these subunits, whether it's the actin subunits or the tubulin, which is the microtubule subunit, to reconfigure and make different kinds of cytoskeletal arrays as the cell needs. So in terms of the actin cytoskeleton, the building block is G-actin or globular actin, very conserved. From yeast to mammals and even in plants, the actin structure is really sort of almost superimposable to the one shown here from animals. So there is a polarity to the actin subunit. So actin subunit, by the way, binds to ATP. So you can see there's this ATP binding pocket, and that side of the subunit is referred to as the pointed end, or will face what's called the pointed end in the polymer. And the opposite end is the barbed end, or faces the barbed end. And the barbed ends quite frequently face the plasma membrane. So and I'll introduce you where this terminology came and talk about the experiment very briefly in just a second. But there is a polarity. The subunit is not just sort of, a sphere. There's two ends to the subunits, right? And this becomes important because these subunits assemble or polymerize in a head to tail manner. So they don't just bind to each other in any arbitrary orientation, but they bind to each other when they polymerize. So in this picture here, each of these colored structures is a G-actin subunit. So they're all the same. So the color is just helping us appreciate how they're associating with each other. But all the G-actin subunits are essentially the same. And they are binding in an oriented or head to tail manner. Now, the way they bind, it turns out, is with a slight skew, so that gives rise to a helical pitch, and we won't have time to talk about the kinds of helical pitches that are found in actin filament, not important for today's purposes. Here, mainly, I want to emphasize that the head-to-tail assembly of the actin filament gives rise to a polarity for the overall filament, right? So that all of the G-actin subunits are binding such that their ATP binding site is facing the same way for all the subunits. So that ends up being the pointed end, and the other end is the barbed end, right? So there is a polarity to the actin filament. So here's some experimental evidence for that, and actually the basis for this pointed end and barbed end terminology has to do with using the motor domain of myosin, which is skeletal muscle myosin, myosin 2, which binds to the actin filament, right, as part of the sarcomere contraction process in muscles. And so if you just take the myosin motor domain, so here, if we look at panel C here, so one of these sort of highlighted versions is that myosin motor domain bound to a G-actin on the actin filament. And then you see lots of other myosin motors are sort of saturating this filament. So you notice that these myosin motor heads, they bind at an incline, right, to the subunits. So when you saturate the actin filament with these myosin motor heads, you get this sort of arrowhead like pattern, binding pattern. So in A, I don't know if you guys can see that on your screen, you can sort of just about see these sort of arrowhead structures, and here is a cryo-EM rendition in B, and C, you're looking at some structure represented in ribbon diagram. And so this is the origin for the pointed end and barbed end, right? So there's what looks like the pointed end of the arrowhead and the barbed end of the arrowhead. So remember, from the molecular perspective, the pointed end is where the ATP binding site is in the G-actin subunit, and the opposite end is the barbed end, okay, or will be facing the barbed end. Now, why is this important? So the polarity, the structural polarity of the actin filament plays a huge role in the kinetics polarity or asymmetry of the actin filament elongation. So here, what they did in this experiment was they assembled an actin filament in vitro in a test tube. And because this is a self-assembly process, you can drive assembly by just packing a lot of subunits, right? So if you have a high concentration of actin subunits, they will by chance bump into each other in the right way that mimics an actin filament, and then you have a seed, which is a primer, and that will grow to form a filament. So they took a preformed actin filament, decorated it with the myosin motor protein, so you can visualize the orientation of the filament, right? So you can see now that's the pointed end because all those arrowheads are facing upwards, and the bottom is the barbed end. They took that decorated seed, where now you can visualize the polarity of that seed under electron microscopy. This is negative stain EM. And then they added additional G-actin subunits, right? So let that polymer grow. They let it grow for some period of time, and they took some of that from the test tube and visualized them using electron microscopy. And you see that there's a huge difference. There's an asymmetry in the growth at the two ends, right? So the barbed end grows a lot more, so you can appreciate that. So the undecorated part is the new growth, right? And compared to the pointed end. So there's a big mismatch in terms of the growth rate between the barbed and the pointed end. I'm not going to go into the kinetics in detail. The main ideas I want you guys to get is actin polymerization rates are very different between the pointed end and the barbed end. And this has to do with what's called the critical concentration, which is the threshold concentration of subunits that you need to drive assembly. And it turns out that the critical concentration for actin addition is much lower at the barbed end. That's the one that I've circled here compared to the pointed end, okay? And so it's, so the actin subunits bind more effectively, add on more effectively at the barbed end compared to the pointed end. It turns out that the actin subunits like to polymerize primarily when they're in the ATP state. Okay, that's a detail that we don't really have to worry too much about for today. So that's really just a very brief introduction to the actin subunit and assembly. In cells, actin filaments are assembled and into a variety of different structures, and cells can have very different types of actin structures, that locally, between just adjacent regions, you could have a filopodium and then something different, like a lamellipodia type structure adjacent to that. And so cells can very carefully control the dynamics as well as the assembly of the actin filaments to form very different types of structures. And this occurs, the mechanisms for that are lots, literally hundreds of actin binding proteins, or ABPs. That's the acronym that you might see in the field. if you look at any of the literature in this field. And these hundreds and hundreds of actin binding proteins regulate various aspects of actin assembly and dynamics. They regulate the nucleation of actin, when and where an actin filament will form in a cell, how long it will grow, whether it's going to be cut or cross-linked, or whatever it is. There's all aspects of sort of actin formation are carefully controlled and fine-tuned in cells. And this picture here just gives you a brief snapshot of some of the major families of actin binding proteins. There are some proteins that bind to the monomer itself, like profilin is a famous one that sort of promotes, enables those actin filaments to polymerize by facilitating nucleotide exchange. So it essentially keeps the actin subunit in an ATP state, which is the polymerization competent state. There are other actin binding proteins that cap actin filaments. So they prevent further addition or removal of subunits. So they sort of freeze the polymer. Okay, if you cap the filament on both ends, that polymer is now stable and no longer dynamic, right? Very important, for example, in muscles. You don't want actin filaments in your sarcomeres of the muscles to be growing and shrinking. That would cause all kinds of problems for muscle contraction. So you fix the filament length using capping proteins. There are other proteins that will cross-link actin filaments in a variety of configurations into very tightly spaced linear arrangements or more net-like arrangements. There are also proteins that will cut actin filaments, so severing proteins like cofilin and so forth. So lots and many other proteins that are not even shown here. But these are the main activities that, the ways that cells control what an actin filament is doing or when it is formed in a cell. So this is how cells then generate this really large diversity of actin structures. And so here you don't have to worry about any of the details. The purpose of this slide is to show you that different cells express different types of actin binding proteins. And so this enables them to construct different types of actin arrays. So Liz Haswell briefly mentioned the stereocilia on the ear cells. So you can see that those finger-like projections, and the same in microvilli on the epithelial cells lining your gut, they're actually shown here in B. You have these finger-like projections that are maintained upright through actin bundles that are within this filament. So a good example of how bundling of actin is really important to provide rigidity or stiffness to maintain these projections, right? If you used microtubules as a thought experiment to make these projections, you would need fewer microtubules to maintain that structure because of the inherent stiffness of the microtubules compared to an actin polymer. Okay. And lastly, just nucleators of actin filaments. There are more than these two that I'm showing here, but these are the sort of most famous ones and the ones that you'll most likely encounter. There are two types of, two major nucleators of actin filaments in cells. One is formins. So formins mediate unbranched actin filament formation as well as promote their elongation, form the barbed end. I won't go into any of the details. Formin is a dimer, it sort of is a donut-like structure, has lots of profilin binding sites, so it sort of attracts a whole bunch of actin subunits and binds to two actin subunits. So it basically, it binds to two actin subunits, each monomer binds to an actin subunit, and by virtue of clustering or concentrating actin subunits in the vicinity, it sort of promotes the growth of that polymer also. Okay, and it's a very processive enzyme. So it sits at that growing barbed end. Conversely, you have the ARP2/3 complex that mediates branched actin filament nucleation. Okay, so you'll see lots of examples in other lectures also, and in your research where you have unbranched or linear actin filaments and then branched actin filaments. And you see linear actin filaments like stress fibers or others, you should be thinking formin-like mechanisms, and branched actin filaments like an lamellipodia would be an ARP2/3 complex-based mechanism. And so here, the ARP2/3 complex is the name for that is it consists of these two proteins called ARP2 and ARP3, which resemble the actin subunit. So it's actin-related protein. That's what the ARP stands for. So the ARP2/3 complex consists of ARP2, ARP3, and 5 other proteins, I think, if I remember correctly. to form an ARP2/3 complex. There's other mechanisms that regulate when the complex is active and so forth that we won't have time to go into. But once activated, the ARP2/3 complex docks binds to a pre-existing actin filament, perhaps one that has been formed by a foramen or another ARP2/3 complex, and then will initiate a new actin filament. So you have a mother filament and a daughter filament, which is the new one that is generated by the ARP2/3 complex. Okay, any questions so far? This is, I think, yeah, that's the last slide for the actin cytoskeleton. All right, so microtubule cytoskeleton. So EM images, the most striking images are the ones in cross section where you can appreciate that the microtubule indeed is a hollow, excuse me, cylinder structure. And you can see these little bumps, and there's 13 of them typically, and I'll talk about what those are. Those are sort of individual protofilaments that associate to form the full cylinder. So again, we've talked about this. Microtubules are relatively stiff, non-compressible polymers compared to actin filaments. The building block for microtubules is a heterodimer. So actin, the building block, is a monomer, G-actin. For microtubules, it's a heterodimer consisting of alpha and beta tubulin. So these are alpha beta tubulin form an obligate heterodimer. So that's how they exist in cells. Okay, there's a very elaborate chaperone machinery that is responsible for assembling these correctly. But this is what the heterodimer looks like here, right? So space-filled version and then this ribbon version here. Both alpha and beta tubulin bind to GTP. So actin is a ATP binding protein, tubulin is a GTP binding protein. Now, although both bind to GTP, the alpha tubulin GTP, the GTP that is bound to the alpha tubulin. So you can see that the GTP binding is right there, and the beta tubulin binds GTP in a similar position. The structures you can see for both alpha and beta are quite similar, and also very conserved protein. Again, if you compare tubulin structure between plants and animals, very similar. Okay, some few differences, but overall structure is highly conserved. The alpha tubulin GTP is non-exchangeable. So it is just stays in that GTP state. Okay, and that's because it is sandwiched between the alpha and beta, and so it is not exposed to the cytoplasm and therefore is not exchangeable. Okay. The beta tubulin GTP sort of faces the cytoplasm, isn't covered up by another subunit, at least to begin with, and therefore the beta tubulin GTP is exchangeable. So when you hear people talking about the GTP state of tubulin subunits or microtubule, when the subunits have been incorporated in a microtubule, that is always, always, always referring to the beta tubulin GTP. The alpha tubulin GTP is boring, nothing happens. It binds to GTP and that's the end of that, right? The beta tubulin GTP is the one that's doing the interesting stuff of hydrolyzing and getting converted to GDP and then being exchanged for a fresh GTP. So here's again just a very brief view of microtubule structure. The subunits, the tubulin heterodimer subunits, just like we saw for the actin filament, assemble in a oriented or head to tail manner. So again, giving rise to a polarity. So here it turns out that the beta tubulin end is the one that is what's called the plus end, the more dynamic end, and the alpha tubulin end is referred to as the minus end. The plus and minus end have nothing to do with charge. Okay, it's just terminology that denotes one end versus the other end, and the plus end, by definition, is the more dynamic end of the microtubule. Happens to be the beta tubulin facing end. So don't confuse this with a charge of any kind. Okay, there are charges, but that has to do with the C terminus of tubulin, etc. But not the plus and minus end of the microtubule is not referring to charge. Now, if you look at the structure of the microtubule, the actual cylinder structure of the microtubule, you can see in panels C and D, they consist of these individual linear filaments that are called protofilaments. So I've highlighted one of them here at A. So there's 3 protofilaments shown here. You can see they're associated longitudinally, laterally, right? Yeah. So typically in most cells, there are 13 protofilaments that associate laterally to form a microtubule. You can have other variations, particularly in a test tube. You can control conditions to form microtubules that have 13 or 15 or even more protofilaments, but in most cells, it's 13 protofilaments that come together to form a microtubule. And just like the actin cytoskeleton, the polarity of the microtubule matters. As I've already said, the plus end is the more dynamic end. And it's the same reason. The critical concentration for tubulin is lower at the plus end than at the minus end. And therefore, tubulin tends to bind more readily at the plus end. So the plus end grows faster, right? So and tubulin likes to bind in the GTP state. Again, this is all beta tubulin GTP, right? Alpha tubulin is always GTP. Now, something weird that microtubules do is what's shown here. So in most cases, you would assume that, if you have a lot of subunits, unpolymerized subunits, so you have lots of Lego bricks, you now have the capacity to build more, right? Polymerize more. And if you have limited number of Lego bricks, then you're limited in terms of your growth. And in most polymer systems, this is the case, right? So for F-actin or filamentous actin, they will grow as long as there's a sufficient amount of G-actin around, all other things being equal, right? But microtubules don't behave this way. Even in the presence of abundant tubulin subunits, microtubules will very abruptly switch from a growth phase to a shortening phase, and then abruptly go back from into the growth phase. So they sort of stochastically sort of toggle back and forth between polymerization state and depolymerization state. And this non-equilibrium behavior of microtubules is called dynamic instability. Okay? It's a hallmark of microtubules. So here you can see what this looks like in vitro. So this is on the bottom is an axoneme derived from a cilia that sort of acts as a nucleator to initiate a polymer formation. There's a single microtubule growing. The free end is the plus end. So you can see the polymer grows, it's lengthening, and then will suddenly switch to shortening, and then it can re-switch back to lengthening and so forth. Okay. So the terminology that is used to describe this, sorry, the Zoom window. Oh, yeah, I have included the key on the bottom. I'm not going to waste our time right now, but rescue refers to a transition from shortening to growth phase, and catastrophe refers to the opposite, a transition from growth to shortening. I included those here just in case you encounter them somewhere as you're reading a textbook or a paper. The model that is proposed to explain why microtubules exhibit dynamic instability is called the Gtp Cap Model or Gtp Cap hypothesis. Okay, and what it states is essentially, remember, the tubulin subunits polymerize in a GTP state, right? So when the beta tubulin is bound to GTP, that's when the tubulin subunit has a propensity to associate at a growing end, be it the plus or minus end, to elongate that polymer. So that's what you see here, right? So the tubulin, the green indicates a GTP state tubulin, the red, yellow indicates GDP state tubulin, right? So tubulin comes on, bound to GTP, and after polymerization, that GTP is hydrolyzed to GDP, right? So that beta tubulin now is in a GDP bound state. And turns out that the GTP bound tubulin has what's called a straight conformation. So you can see that red dotted line shows that those alpha and beta tubulins are in register, right, in this dimer. When you go to a GDP bound state, where that beta tubulin is now in the GDP, where the GTP has been hydrolyzed to GDP, you get this curved conformation where the beta tubulin sort of lists backward. Okay, so GTP tubulin has a straight conformation, GDP tubulin has this curved conformation. And so in a microtubule lattice, so you can see there's very little GTP tubulin that's really restricted to the tip of the microtubule, and the bulk of the polymer is GDP tubulin. So there's a lot of stress that's built up in this lattice because all those GDP tubulins are sort of bending outward or trying to bend outward. All the lateral associations between the neighboring protofilament tubulins and longitudinal associations between the tubulins within a protofilament are sort of helping keep things together, but they're all sort of want to sort of bend backward. And so the idea of the GTP cap hypothesis is that it proposes that so long as you have a layer of GTP tubulin at the growing end or at the plus end, that stabilizes that polymer and keeps it in a growth state. if you lose that GTP cap, and that loss could be partial too, as you can see in this picture here. So not, there's some GTP tubulins, but not all protofilaments. Then that leads to a switch to depolymerization, right? And so the microtubules will abruptly go from polymerization to depolymerization. And so this is the hypothesis that sort of is used to explain why you have this dynamic instability behavior, even in the presence of tubulin subunits. Right? Any questions about this?
I have one really quick.
Sorry, I didn't catch that.
I was just wondering if there was a way for the microtubules to be in a state of being static. Does it have to either be elongating or kind of breaking down?
Sorry, that got quite broken up on my end. I couldn't catch that. Can you state it briefly? Just what's the main part that you want to know?
Is it possible for the microtubules to be in a state of static? Does it have to constantly be elongating or breaking down?
No. So cells, again, so that's basically a great segue to this slide, right? So again, cells control the behavior of microtubules just like they control the behavior of actin filaments. And there are post-translational modifications that can occur on the tubulins that are part of the microtubule polymer, as well as other proteins. And those microtubule binding proteins are called microtubule associated proteins. So there should be a hyphen between microtubule and associated, or MAPs. And so again, there are hundreds of microtubule associated proteins that control all aspects of the life of a microtubule. Nucleation of the microtubule, their elongation from both ends, whether they are cross-linked, whether they are severed or cut, and so on and so forth. And so there are ways in cells to stabilize microtubules. So a good example of that is in a cilia or flagella, that beats, and I'll introduce some of those toward the end of the lecture. And their microtubules are in very stable. And that's because of post-translational modifications and other mechanisms that stabilize them. Yeah, good question.
Thank you.
So again,
quick question.
Yeah, go ahead.
Yes, it looks that the microtubule inside is empty, and what is the material inside of the microtubule?
Yeah, that's a long-standing question. I would say unclear still. There have been some papers recently that I actually haven't had a chance to read through in detail. There was something I saw recently that said that there are things inside the microtubule, and whether they were small proteins or other components, I don't remember now. But that, I would say, certainly is still not something that's nailed down or sort of well accepted or widely accepted. Yeah.
Hey, Ram, can I chime in on that?
Yeah.
Yeah, so I mean, one of the ones that comes to mind, right, is the acetylating enzymes.
Right. Oh, yes. The acetylating enzyme is, go ahead and talk about that.
No, I was just going to say that, one of the common post-translational modifications of microtubules that can affect their stability, can affect their flexural rigidity, is acetylation, which occurs on the luminal face of the microtubule. And it's believed that the acetylating enzymes actually can diffuse within the microtubule to acetylate it along its length. So there is some activity that can be important for microtubule structure that does occur within the lumen.
But have people demonstrated the diffusion inside the microtubule? Because microtubules can be
that's
pretty long.
That's still a controversial point, Ram. And so there's one idea that it gets in through holes in the lattice structure. So if you have a loss of tubulin dimers from the lattice, the enzyme it could get into the microtubule that way. Others think it comes in through the plus end. There has been some evidence of very slow diffusion through the microtubule, but I'd say it's still an open question.
Right. Yeah, acetylation is definitely the example that I mentioned in my class, as well as most microtubule post-translational modifications happen on the outside and on the C-terminus, which is what, the tubulin that face the outside of the microtubule. The one famous example where it happens luminal or inside the microtubule cylinder is the acetylation one that Ben is talking about. Ben, did you happen to come across this paper? There was something I saw on PubMed recently about evidence about this XYZ inside the microtubule, that they were like, oh, look, there are things inside the microtubule. I don't know if you came across that or not.
I'm not familiar with the
okay, paper. That's fine. I'll look it up. If I find it, I'll email it to you guys. But yeah, I would say it's still a very open question. There have been some weird ideas about quantum consciousness and this and that and the other that have to do with microtubules and what's on the inside and whether they're charged or not. And that's still, you know, has strictly still remain in the realm of sort of the unusual and not accepted.
And also the radius is very small. So if it's a fluid, whether the surface tension will affect the mechanical property of the microtubule.
Good question. I don't know the answer to that as well. Again, I don't know if you think of the microtubule as a pipe, right? So then you have this, what is that called? The boundary effect also, right? So I don't know how that would affect things, even fluid motion inside the microtubule.
Okay, I have another question if
Go
you
ahead.
don't mind.
Yeah, that's fine.
Okay.
I don't care if we get through all the slides. I'll be emailing, or patients will be emailing the PDF of this. So you'll have all the information. So go ahead.
Okay, so you can see that the microtubule might buckle. So what will happen after the buckling?
Sorry, say that again. Microtubules will what? Buckle?
Yes.
So microtubules can buckle, and Ben was referring to one of the ways that cells can sort of control that. So post-translational modifications of microtubules, as well as certain microtubule binding proteins, when they bind to microtubules, can soften or change the flexural rigidity of microtubules. And when microtubules buckle, one of the consequences that I'm aware of buckling of microtubules is that they seem to be more prone to getting cut by severing proteins like catanin.
Oh.
So at the point of maximum curvature, there's not a lot of evidence. There's some biochemical experiments that have demonstrated that, and some of my own work that still remains published from my postdoc days. I observed that under conditions when microtubules start to bend and buckle and form sort of these really sharp bends, they seem to break at the point of maximum curvature. Now, I don't know if they're breaking just mechanically, just physically because they bend so much, or my suspicion is because of severing proteins that like to bind at these sort of bent sites that perhaps expose some binding sites at those bent or curved regions of the microtubule.
Okay, thanks.
Mm-hmm. Any other questions?
I had a question too, actually.
Who is this?
It's Kirstine from Merlin.
Oh, okay, yeah. Mm-hmm.
Yeah, so I read that microtubules shuttle things inside the cell. And so I read also that it kind of acts as a conveyor belt. So how does that work? I initially thought that it shuttles things within like its structure because it's almost shaped like a pipe. So does that just actually happen on the surface of the microtubule? Right,
so shuttling things inside the microtubule, as we just discussed, there's really no strong evidence for that at all. There's really no evidence for that, I would say. You know, there are things that happen inside the microtubule. Certainly, acetylation is very well known as a modification that occurs that's luminal. But shuttling of things inside the microtubule, I haven't seen any strong evidence for that. What you're probably referring to is treadmilling. So that's when subunits are added on one end and are being lost from the other end. Actin does this too. And so there's this flux of subunits from one end to the other end. And this is very important in the spindle, for example, part of how the kinetochore microtubules, for example, behave in a spindle apparatus. That's maybe what you're talking about. Now, the bulk of the microtubule dependent transport of material in a cell is motor proteins driven. So this is all kinesins and dyneins, cytoplasmic dynein driving that movement.
All right, awesome, thank you.
Hey, this is Stephanie. I was just wondering, I know you mentioned that there is a polarization along the length of the microtubule. I was wondering, like, for the individual dimers that make up, that pipe structure, is there like a rotational polarization where like the inside portion of the microtubule is, the same?
Oh yes, absolutely. The subunits, the C terminus of the tubulin subunits are all facing the outsides, and the C terminus of the tubulin. So the tubulin you saw is a very globular structure, right? Nice compact protein. Mm-hmm. The C terminus is floppy. It's sort of like a little, brush, like little tail that wags on the surface of the microtubule. So the microtubule surface is sort of hairy and fuzzy because of all the C termini. And the C termini are really important. They're charged. There's lots of glutamine, so that gives the microtubule surface a net negative charge. It's important for a lot of microtubule binding proteins or microtubule associated proteins bind to microtubules through interactions with that C-terminus. So you could cut away the C-terminus using proteases, and now a lot of microtubule-associated proteins then have trouble binding microtubules, for example. So yes, there is a orientation, both inside, outside, and top, top to bottom.
All right, thank you.
Okay. All right, so I'll go through this again. The purpose today is not to go into details. I just wanted to mention this because this is an important complex, which is the microtubule nucleation complex. It consists, again, multi-protein complex. One of the components of this complex is a version of tubulin that's called gamma tubulin. So there are several different versions of tubulin. The alpha beta tubulin are the ones that polymerize to form a microtubule polymer. Gamma tubulin is another version that is part of the nucleation complex, so which consists of gamma tubulin, these yellow proteins here on the bottom, along with other proteins that are called gamma tubulin complex proteins, or GCPs. And this is the complex that initiates microtubule nucleation in cells. There are other versions of tubulin as well that are found in cilia and flagella that we don't have time to talk about today, epsilon, delta tubulins, and so forth. So there's a handful of different tubulin types. But this is the main, really the sole sort of microtubule nucleator. So there's no like formin, ARP23, there's different kinds for actin, there's spire and others as well. But here for microtubules, you have the gamma tubulin ring complex as the sort of primary microtubule nucleation complex in cells. Next, intermediate filaments. So I'll go through a little bit quickly here. I want to really emphasize more of the differences. So unlike actin and tubulin, the subunits that make up intermediate filaments vary considerably in size. So you can see that neurofilaments, these ones that say NFM or whatever, the different types here are very large in terms of their protein size compared to lamins or some, even fermentin for that matter. And also intermediate filament subunits do not bind to nucleotides. So actin binds to ATP, tubulin binds to GTP, but intermediate filament subunits don't have nucleotide binding. Intermediate filaments, the other important distinction is that the intermediate filaments lack polarity. And this picture on the bottom sort of shows you a little bit about the hierarchical process through which intermediate filaments assemble to form the final 10 nanometer or so intermediate filament structure. But the assembly unit is a tetramer, and within the tetramer, the two dimers that make the tetramers are facing opposite directions. So that assembly unit has no net polarity, and therefore the intermediate filament polymer itself has no net polarity. Okay, so it's an apolar structure. I'm going to skip this slide just to quickly touch on, in the last closing minutes, on how the cytoskeleton plays a role in cell morphogenesis in animals and plants, and then I'll wrap up with a very brief discussion of motor proteins. And if we run out of time, that's fine. Feel free to email me and ask questions once you've seen the slide deck. Patients will have emailed that to you if she hasn't already. So the cytoskeleton plays a very direct role in shaping animal cells. Our cells are soft and squishy, and the forces exerted by the cytoskeleton, whether they're pushing or pulling forces, can deform the plasma membrane and thus changing the cell shape. In plant cells, that doesn't work, right? Because plant cells, have a very tough cell wall surrounding them. And as Liz mentioned yesterday, there's also this incredible turgor pressure inside that can be 5 atmospheres. That's 3 to 5 times your car tire pressure. Okay, just to give you some context, that's pushing out, and the cell wall is resisting that, right? So you take away the cell wall, cells explode under that turgor pressure. So because cytoskeleton generated forces pale compared to the turgor pressure and cell wall stress, the cytoskeleton in plant cells plays an indirect role in shaping cells. So how does this work? So in animal cells, like I said, the cytoskeleton generated forces can push and extend the plasma membrane, or pull and retract the plasma membrane and change the shape of the cell, important during cell migration, for example. But in plant cells, what happens is that the turgor pressure which is really hydrostatic pressure. So the plants just accumulate a lot of solutes and build up that osmotically this hydrostatic pressure. And hydrostatic pressure is isotropic, right? It's the same in all directions. So you'd expect the plant cell to simply balloon, right? As the turgor pressure grows and pushes and drives the growth of the cell, it would just sort of be spherical. And that's what happens if you digest carefully under isotonic solution conditions, the cell wall. If you digest over the cell wall, the cell, perfect sphere. But instead, what plants do to harness that turgor pressure and convert that into cell shape is they use the cytoskeleton to structure the cell wall material. So the cell wall material isn't deposited randomly, but it is deposited in an asymmetric or anisotropic manner. So if you have cell wall materials like cellulose, which is a very strong material, pound for pound, almost tensile strength is comparable to that of steel. If the cellulose fibers are wrapped around the cell, this orientation, then it resists growth in that axis, and now the cells grow in an orthogonal manner, right? So this is how you channel a turgor pressure into a directional growth in plant cells. Where does the cytoskeleton come into the picture? Well, the cytoskeleton in the cell acts as a scaffolding structure that guides deposition of cell wall material. So that oriented deposition of cell wall material is occurring because of the cytoskeleton scaffold. And in plant cells, the main one is the microtubule. Animal cell morphogenesis, a lot of it has to do with actin cytoskeleton, causing lamellipodia, filopodia to form, cells to migrate, and so forth. Plant cell morphogenesis is primarily a microtubule driven process. Actin is important as well, but you depolymerize the microtubule cytoskeleton using drugs, for example, cells balloon. Okay, you depolymerize the actin cytoskeleton, there's a little effect, but not a huge effect. So you can see in this picture, the microtubules in plant cells sort of are plastered to the plasma membrane, so they are right beneath the plasma membrane in plant cells, and these microtubules guide the oriented deposition of cell wall material like cellulose. And so if the microtubule is oriented in this direction, the cellulose becomes oriented in that direction, and then the cell grows long, as I showed on the previous slide. If you change the microtubule orientation, you will change the cell wall deposition. The new material that's being deposited will have a different orientation, and that will affect the growth of the cell, the growth axis of the cell. Okay. So the plant cytoskeleton does not serve fundamentally as a supporting structure because it's just too soft, too, in the face of the cell wall and the turgor pressure, but really is a scaffolding material that's indirectly conferring cell shape. Does that make sense? I'm going to skip that. It has to do with plant cell expansion that can be either sort of more or less uniform along the surface of the cell, or it can be focused at a particular point, and that gives rise, only that tip is then growing. And this is also seen in animal cells. So for example, growth cones and neurons are sort of a tip growing process where only that tip is growing. Okay. Same thing in plant cells. All right, cell migration. So here the cytoskeleton again plays a big role. The velocity of cell movement can vary over many orders of magnitude. The winners are the ciliated cells. So sperm can move at velocities of hundreds of microns per second. Tetrahymena, which is multiple cilia, moves a little faster. And amoeboid motility in general is much, much, much slower, except for these big amoeba, the amoeba proteus that you see in sort of high school experiments and things like that. They are large and they can really extend big pseudopods and can move at reasonable rates. But in our immune cells, like white blood cells or like leukocytes and fibroblasts, they crawl at much slower speeds. Right? And nerve cells are really, really slow because it's not just the growth is slow because of amoeboid, but it has to do with perceiving growth factors and making sure the nerves are growing in the correct orientation, making correct connections and so forth. So that's a slower process. They're not just sort of growing or migrating, right? Randomly, it's a directed process. Okay, so amoeboid cell motility is here are the take-home messages, really actin-driven process, all right? So the famous model that Tom Pollard used to show a long time ago in many, many, many conferences was this dendritic actin nucleation model. The idea is pretty straightforward. And I'll just describe that. You don't have to worry about any of the molecular details here. Okay, so ignore this busy, crazy looking picture. It's actually a pretty interesting and really informative picture when you look at it and think about it. But the basic idea is, if the cell wants to extend in one direction, it needs to exert pushing forces on the plasma membrane to extend that plasma membrane in that direction. And that happens by activating R2-3 complexes locally in that position, in that region of the cell. And lots of R2-3 complexes get activated, and they will dock on to pre-existing actin filaments and initiate new actin formation, right? So you can see in this EM picture in the inset here, this huge, just really incredibly dense branched actin network, right? So that's all due to all those R2-3 complexes like forming new branched filaments. But actin is floppy, can't really push effectively unless, if it grows long, right? Because it will just buckle and bend and become floppy. So there are capping proteins that cap those newly nucleated actin filaments to keep them short. So you have lots of, hundreds of thousands or more short actin filaments now growing against the plasma membrane. The barbed ends are facing the plasma membrane. And all of those are then exerting that pushing force that extends that edge forward. Okay. But if you want to keep that extension going as the cell migrates, you need to feed a lot of actin subunits, right, to maintain that growth state. And so that works by recycling the actin subunits. That's what's happening on the back end here. Okay, so here on the top, you see lots of actin nucleation and capping to keep those filaments short. And then on the back end, as the older filament gets chopped up, and those subunits get recycled to can maintain or sustain that continued polymerization at the growing end. Okay, so this is how you have this amoeboid motility, where you push and extend the plasma membrane, cell adhesion proteins then adhere to the extracellular matrix typically to keep that extension stapled to the surface, and then you retract the back end, and that's typically a myosin-driven process. So this is how our cells move, and cells can then sense information and change that direction of movement, right? There are great videos if you look on YouTube of neutrophils chasing bacterial clusters, for example, a very good example of how cells can rapidly change their direction of migration in response to external cues. So cilia-based motility, a lot faster. And here, the hardware is really microtubule based. Okay, so cilia or flagella are really the same thing in terms of how they're put together. They're complex structures. They're really organelles. There are hundreds of proteins that make up the cilia and flagella. They have this 9 doublet microtubules in the periphery and 2 pairs of central microtubules of lots of other elaborate structure. But these cilia and flagella beat in specific patterns, and that's what propels the cell in fluid environments. Okay, and finally, we are actually out of time. So I am going to just all I'm going to mention here is that the cytoskeleton so far have really emphasized their role from a structural standpoint, right? From a mechanical standpoint, exerting forces or organizing cells or conferring cell shape, but they also serve as tracks for motor proteins. And you have actin-based motors, they're called myosins, and there are microtubule-based motors that can be either kinesins or dyneins. Myosins and kinesins are evolutionarily related. Dynein is a whole different type of protein called AAA ATPase. Nonetheless, all of these motors use ATP for fuel. So they're all ATP dependent enzymes. And kinesin 1, the conventional kinesin, is the smallest, most compact motor protein. Really has a very high efficiency even better than our automobiles. And so I will leave it at that. And like I said, again, you'll have all these slides. I just had 2 or 3 slides at the end here about motor proteins. And they're pretty straightforward, so you're welcome to go through those. And if you have any questions, please email me at any point. So I'll stop there and take any questions. And we can also, if you don't have any questions, feel free to mute and leave for 5 minutes, and then I'll introduce Ben.